Perfect. So hi, everyone. My name is Daphne, and I'm a third-year PhD student at the University of Calgary. And today I'm going to be talking about brain iron and anxiety symptoms in children with and without prenatal alcohol exposure. Okay. Before I get started, I would like to acknowledge that I'm joining from the traditional territories of the Treaty 7 region and Métis Nation of Alberta, Region 3, which some of you may know as Calgary, Alberta. As a settler, I'd like to give my thanks to the Indigenous peoples of Canada who have cared for this land and continue to fight for environmental rights. So prenatal alcohol exposure, which I'll be referring to as PAE, can cause a broad range of physical, mental, and behavioral deficits that persist across the lifespan. 10.8% of women in Canada report drinking during pregnancy, and this can result in a diagnosis of fetal alcohol spectrum disorder, FASD. And here you can see the 10 functional domains that are assessed during an FASD diagnosis. And this really shows how heterogeneous the effects of PAE can be. Interestingly, individuals with PAE who do not meet diagnostic criteria for FASD report experiencing similar problems to those that are diagnosed with FASD. So today I'm going to be focusing on two of these domains, the neuroanatomy as well as affect regulation. And in the diagnostic criteria for FASD, a diagnosis of anxiety or depression disorders fits into this affect regulation category here. So in children and adolescents, mental health problems can be thought of as internalizing or externalizing symptoms. Internalizing symptoms refer to negative behaviors that are reflected inward, and they're often related to mood and emotions. So associated disorders would be depression or anxiety. And then we have externalizing symptoms, which are negative behaviors directed outwards. And these are often related to impulsivity and aggression. So associated externalizing disorders would be attention deficit hyperactivity disorder, ADHD, as well as conduct disorder. So more than 90% of individuals with FASD have a co-occurring mental health problem. And in individuals with FASD, depression is 4 times more likely than in the general population. Anxiety is 11 times more likely than in the general population. And in externalizing disorders, ADHD is 10 times more likely than the population. Oppositional defiant disorder is 13 times more common in those with FASD. There we go. Okay. So brain iron is really important for healthy brain development. And you can see on this image on the right that iron is involved in neurogenesis, myelination, and neurotransmitter synthesis during the prenatal and early postnatal period. And where the red is darker in this image is where iron is most important for each specific process. So postnatal brain development relies on fetal stores from the fetal iron stores from the 3rd trimester, because iron is not fully regulated to cross the blood-brain barrier yet. However, we know that alcohol during pregnancy lowers fetal brain iron, and therefore brain iron may be affected in children and adolescents with prenatal alcohol exposure. So Kim and Wesling-Resnick have a review paper where they talk about potential mechanisms of how iron can affect emotional behavior. And I've adapted this figure to highlight, this figure from their paper to highlight some of those mechanisms. So these are based on basic research, but there's quite a bit of evidence to suggest that monoamine and neurotransmitter synthesis and metabolism are affected by abnormal brain iron. So how do we measure brain iron? One way to do this is by using quantitative susceptibility mapping, QSM. And QSM is an MRI technique that measures tissue susceptibility. And I know we've heard a lot about resilience and susceptibility throughout this conference, but when I'm talking about susceptibility, I'm referring to magnetic susceptibility as an indicator of brain iron. So the higher the susceptibility indicates more brain iron, and negative numbers indicate less brain iron. So subcortical structures are rich in non-heme iron. And so we know that iron is the main susceptibility source in these structures. And on the right, you can see a QSM image. So the bright areas are just in this figure is the pallidum. And it, as you can see, it corresponds with a positive number in the color bar, indicating that it is an area with high iron content. So brain volume of basal ganglia and limbic structures are predictive of depression in adolescents. And we often see that children and adolescents with prenatal alcohol exposure also have small brain volume and basal ganglia and limbic structures. However, we only have seen one study that's found a trend level association between anxiety and caudate volume in children with PAE. So we don't really know if brain volume in individuals with PAE is also associated with mental health problems. And then we also know that low brain iron in the thalamus and putamen are associated with ADHD, and high brain iron in the thalamus and putamen are associated with depression symptoms in adults. But we don't really know if brain iron is affected in children with PAE, and if this might relate to externalizing symptoms like ADHD or internalizing symptoms like depression. So we wanted to know, is brain iron associated with internalizing or externalizing symptoms in children with and without PAE? And is brain volume associated with internalizing or externalizing symptoms in children with and without PAE? And so based on the literature, we hypothesized that lower brain iron in the caudate and putamen would be associated with more internalizing and externalizing symptoms. And we also hypothesized that there'd be a group brain iron interaction in the thalamus, where we would see a lower brain iron in the PAE group, which would be associated with more internalizing and externalizing symptoms. So we had 59 participants, 40 without PAE and 19 with PAE, aged 7.5 to 15 years old. And for the participants with PAE, 7 had a prior diagnosis of FASD. And as you can see, many of the participants with PAE also had other co-occurring mental health diagnoses. So each participant had a T1 weighted and QSM MRI scan. And with a T1 weighted scan, we're able to extract information about volume, and QSM scans provide information about iron deposition. So the T1 weighted scans were processed through FreeSurfer 6.0, and the QSM scans were processed through MATLAB using a code written by Dr. Hongfu Sun. So we looked at 7 regions of interest: the nucleus accumbens, caudate, putamen, and pallidum, as these regions have high iron deposition. And then we also looked at the thalamus, hippocampus, and amygdala, because these are limbic structures that are often affected in mental health problems. So we had parents or caregivers complete the behavioral assessment system for children, the BASC-2. And the BASC provides T-scores that are age and gender normed. So the T-scores have a mean of 50 and a standard deviation of 10. Scores below 60 are considered developmentally appropriate in terms of behavior, and scores between 60 and 69 are considered at risk of developing a disorder, and scores above 70 are considered to be clinically maladaptive, indicating that a child may meet a diagnostic criteria for a given internalizing or externalizing disorder. And what you can see here is that the internalizing problems composite score includes depression, anxiety, and somatization subscales, while the externalizing problems composite score includes aggression, conduct problems, and hyperactivity subscales. So I ran an ANCOVA for each brain region to assess the association between internalizing or externalizing problems and volume and susceptibility with group and group brain interactions while controlling for age and gender in the model. And for structures where susceptibility or volume was significantly associated with behavior, I also tested associations with internalizing or externalizing subscales. Okay, so to orient you to these figures, you can see on the y-axis that we have t-scores for different scales. And then on the x-axis, it's showing the susceptibility residuals after being controlled for age and gender. And this is susceptibility as in brain iron. And then I also have the results, both uncorrected and corrected in P and Q. So for the nucleus accumbens, you can see that brain iron was negatively associated with internalizing problems, depression, and anxiety symptoms in children with and without PAE. And looking at the far right, this association between brain iron and internalizing problems seems to be primarily driven by brain iron and anxiety in the nucleus accumbens. So when we move on to the amygdala, we see the opposite trend with internalizing problems and anxiety associated with increased brain iron in the amygdala. And then looking at the somatization subscale, we found a group brain interaction. So higher brain iron was associated with more somatization symptoms in the PAE group, but there was a slightly negative association found in the unexposed group. And the dashed lines here indicate a non-significant association within the group. And then again, the anxiety subscale association seems to be driving the internalizing problems association. So moving on to the thalamus, we found a group interaction in internalizing problems, as well as the depression and anxiety subscales. So children with PAE had a negative association between internalizing symptoms and brain iron, while the unexposed group had a positive association. And again, this is really being driven by the anxiety subscale. And then the putamen was the only brain structure associated with externalizing symptoms. And here we see this interaction effect where the PAE group is showing a positive association between putamen, brain iron, and externalizing problems. And the unexposed group is showing a slightly negative association. Interestingly, there were no significant associations between putamen, brain iron, and externalizing subscales. And then there were also no other associations between any other brain regions, including brain iron or brain volume for internalizing or externalizing symptoms. So how do we make sense of this? Well, the nucleus accumbens is part of the mesolimbic dopaminergic system. So it's possible that the nucleus accumbens may be impacting behavior through dopamine metabolism. And in iron deficient rats, there is a decreased dopamine transporter in the nucleus accumbens, which can result in elevated dopamine, leading to desensitization of the dopamine transporter. We also see decreased dopamine 2 receptors in the nucleus accumbens of iron deficient rats. And then there's been really limited research on amygdala brain iron, but it's possible that increased iron in the amygdala could be related to more sensitive emotional appraisal or due to increased stress hormones, but that's very speculative. And then previous research has shown that increased brain iron in adults with depression. And we saw this association in the unexposed group, but the opposite trend in the PAE group. And then lastly, it was surprising that there were no associations between thalamus, brain iron, and externalizing symptoms, given the previous research on ADHD and low brain iron in these structures. So we did see an association with putamen, brain iron, and externalizing symptoms, but again, I would have expected to see an association with the hyperactivity subscale, given the previous studies on ADHD and putamen, brain iron. So to summarize, this is the first study to look at brain iron and mental health in children with and without PAE. And we found that brain iron is associated with internalizing symptoms, mainly anxiety, as well as externalizing symptoms in multiple brain regions. So Group brain interactions in internalizing and externalizing symptoms in some brain regions suggest that the mechanism of how brain iron impacts behavior differs in those with and without PAE. And our lack of findings between brain iron and, our lack of findings between brain volume and internalizing or externalizing symptoms suggest a need for more specific brain measures, such as QSM used here, in order to better understand the neurological underpinnings of how PAE affects the brain and behavior. So I'd like to thank our funders, my supervisors, and lab members for making this research possible. Thank you to the organizers of this conference for the opportunity to speak, and thank you for your time. I'd be happy to take any questions if there is time.
Thanks, Daphne. I think we have time for a quick question if there is one. Anybody have a question? No. Maybe I'll ask just a brief one, Daphne, because I think we get this a lot, but just can you distinguish for folks between PAE and the diagnosis of FASD and just what that means in terms of your population?
Sure. So as I kind of mentioned, in our sample, we had 7 kiddos that had a diagnosis of FASD, and then the other 12 did not have a diagnosis. What we kind of see is that individuals with a diagnosis and those without diagnoses that still have prenatal alcohol exposure do experience this similar difficulties. So I don't know if you saw there on one of the slides, I didn't point it out, but 11 of our participants actually had prenatal alcohol exposure meeting diagnostic criteria, but they have either not had their assessment yet or don't meet the criteria for an FASD diagnosis. So in terms of what this kind of means for the sample, this would suggest that maybe this is not as clinical of a sample as a study that had 100% of participants with an FASD diagnosis.